नमस्ते बिटिया, हमेशा खुश रहो बिटिया. Myself Dr. Arti, Associate Professor, JAYOTI VIDYAPEETH Women's University, Faculty of Homeopathic Sciences. Today we are going with a new topic, that is the Bacillus. Bacillus is a bacteria which having the two species which are very important. One is a Bacillus cereus, which is causing the diarrhea, and second one is a Bacillus anthrax, which is responsible for anthrax. First, we'll talk about the general characteristics of the bacillus. They are the 60 species are there. They are gram-positive, gram-variable bacilli. Some they are large. Most are saprophytic, containment, or normal floras. Bacillus anthrax is a most important member. They are producing the endospore, aerobe. Sometimes they are facultative anaerobe. They are catalase positive, rapidly differentiate from the Clostridium. Bacillus suppuratus are ubiquitous that remain in the soil, water, and airborne. They are thermophilic. They can flourish at extremes of acidity and alkalinity. So that means resistance power is too high. The disease associated with the bacillus are the bacillus anthrax, which is causing the anthrax. Anthrax are three types: cutaneous, GI, gastrointestinal, and respiratory, that is inhalation method. Bacillus cereus are causing the gastroenteritis. Then by bacillus mycosites, which are again causing the opportunistic gastroenteritis. Then theriagenesis, which is again the gastroenteritis causing, and other some bacillus species are also there. The laboratory characteristics of bacillus on blood agar, they are large spreading gray white colonies with irregular margin. They are beta hemolytic. They are differentiating. This is helpful in differentiating the various bacillus species from anthrax, which is not beta hemolytic. The spores seen after several days of incubation, but not typically in fresh clinical specimens. First, we'll talk about the bacillus anthrax. In Bacillus anthrax, they are the spore-forming gram-positive bacilli. They are facultative anaerobe, non-fastidious growth of non-hemolytic colony that are firmly adherent to the agar surface. They are polypeptide capsule consisting of poly-D-glutamic acid observed in clinical specimens. The virulence power is so high that the capsule is present in virulent strain. Virulent strain also produce three exotoxin that combine to form the endemic edema toxin, combination of protective antigen and edema factor, and the lethal toxin, that is a protective antigen and lethal factor. Spores can survive in soil for years. The epidemiology considered primarily this is the herbivore with human as accidental host. Rarely isolated in developed countries, but is prevalent in impoverished area where vaccination of animal is not practiced. Individuals at risk include people in endemic areas in contact with infected animal or contaminated soil, people who work with animal material imported from endemic areas, and military, non-military people exposed to infectious aerosols. There is significant concern that the spores will be used in bioterrorism. This is the first bacteria which is used as bioterrorism. This disease they are causing the cutaneous anthrax, which is very much common, inhalation anthrax, which is again most deadly form, then gastrointestinal anthrax, which is a rare but commonly fatal disease. The diagnosis includes the isolation of the organism from the clinical specimens like papule, ulcer, and blood. The treatment includes the ciprofloxacin as a drug of choice, penicillin, doxycycline, erythromycin, chlorpheniramine can be used. But the bacteria are resistant to sulfonamide and extended spectrum cephalosporin. Vaccination of animal herds and people in endemic areas can control the disease, but spores are difficult to eliminate from contaminated soil. The animal vaccination is effective, but human vaccines have limited usefulness. The epidemiology, it is rare in US, exotic in certain foreign countries. Anthrax spores infectious for decades. Biological warfare experiments happen. Three well-defined cycles include survival of a spore in the soil, animal infection, and infection in humans. This is primarily a disease of herbivorous animal, most commonly transmitted to human by the direct contact with animal product like wool and hair. That's why it is called the wool sorters disease. Also acquired via inhalation and ingestion. There is an increased mortality with that portal of entry. Still poses a threat. Important material contaminated with spore from these countries, like for example bone, hides, and other material, usually encountered as an occupational disease, Veterinarian and agriculture workers are the victim. In epidemiology of the anthrax animal and human host, uh, this is a cycle when person more 20% of the time when contact with contaminated animal product, 95% of the time ingestion of contaminated food product, and 95% of the time it is inhalation of contaminated dust. Historical importance of Bacillus anthrax is very much important as it was the first bacteria seen under microscope. It was the first communicable disease shown to be transmitted by inoculation of infected blood. It was the first bacterium to be isolated in the pure culture. And anthrax vaccine was the first live attenuated bacterial vaccine prepared. Now, clinical presentation of the anthrax include the cutaneous anthrax. 95% of the human cases are cutaneous infection. There is a 1 to 5 day after the contact. Small pruritic non-painful papule and inoculated site papule developed into hemorrhagic vesicle and rupture. Slow healing painless ulcer covered with blackish scar surrounded by edema. Infection may spread to lymphatic via local adenopathy. Septicemia may develop. 20% mortality if untreated. In inhalation, 100% fatal. This is pneumonic. Meningitis may complicate cutaneous and inhalation form of disease. There is a fever, pharyngitis, and neck swelling. In gastrointestinal, virtually 100% fatal. There is abdominal pain, hemorrhagic ascites, paracentesis. Fluid may reveal gram-positive clots. Lab diagnosis include the gram staining bamboo stick appearance, nutrient agar, there is a medusa head appearance, gelatin stab agar, there is inverted fire tree, McFadden reaction is there, direct immunofluorescence test and PCR is also helpful. In treatment, penicillin is the drug of choice. Other antibiotics are also used in vaccine. Laboratory workers, it is controversial. It is given to laboratory workers, employees of meal, handling the goat hair, active duty military member, and potentially entire population of US for the herd immunity. The key features of the anthrax and Bacillus cereus. In Bacillus cereus, there is only gastrointestinal diarrhea, nothing else. But it is emetic type and diarrheal type. But the difference in anthrax and Bacillus cereus are hemolysis negative in anthrax but positive in Bacillus other species. Mortality is negative but other are positive. Gelatin hydrolysis is negative in anthrax but positive in other. Salicylene fermentation is negative while positive in others. Growth of PEA agar is negative in anthrax and positive in others. I hope you understand and enjoy this session. If any query, please mention it in comment box. I will try to resolve it. Thank you so much. Bye bye. Take care.